You gotta know a day. Today we're going to talk about the Meselson Stahl experiment, which showed that DNA has semiconservative replication. This is Matt Meselson and Franklin Stahl. And when I say DNA has semiconservative replication, you maybe have seen something like this where you see a DNA, it unwinds, and if it's replicated or being copied, you're going to keep the old strand and add a new strand. In reality, this is what it looks like. As you're making new DNA, it's kind of this complicated process, but that's what happens. You take the old strand, you add a new strand to it. But we didn't always know that. So what they did in their experiment is they took E. coli, which has DNA, and they had it grow in what was called N15. It's called heavy nitrogen. It's got an extra neutron, so it's a little heavier than normal nitrogen. So took E. coli and had it grow in just this type of nitrogen so that nitrogen was in the DNA. And then they took a sample and they called that generation 0. And as you can see, heavy nitrogen, they put it in what's called a ultracentrifuge, and there's cesium chloride in there. They like to ask questions about cesium chloride. And things that are more dense or heavier are going to move farther down, and then things that are less dense or lighter are going to move further up in the tube. So this N15 DNA is down here. Down here, this is the N15 DNA right away. And then they added N14 DNA, N14 nitrogen. It's lighter nitrogen. They let the E. coli grow in this lighter nitrogen, which gets incorporated into the DNA. And what they found is that after one generation, they found this medium band. It's in between the light level and the heavy level. And as you can see, that's the old strand and the new strand. Old strand and the new strand. This separated out, and then you add the new DNA in there. That's generation 1. So that showed that it wasn't perfectly conservative. You didn't keep all the old and then add new to it. They're mixing together and connecting together. In generation 2, they showed now there's 2 bands. Now there's a light band and a heavy band. And that suggests this here, where we have the old and the new, brand new one. But this, just like this one broke into, this one broke into. So you keep the old and add new. You keep generation 1's old and you add generations 2 new. Same thing here. Keep the old, add new. Keep the old, add new. And that's why we get, this light band, because these are just new bands that are that light nitrogen, and then the medium band, because we have the old with new. So this whole time, you never get these heavy bands, so you never get back the original where it is just 2 olds together. You only get the original bonded to a new band, and then over time you add more and more and more of the light as a proportion. But again, it shows it's semi-conservative because it's keeping part of the old and it's conserving the old, but it's adding new. And each time it does that. This experiment that was in E. coli, which is a prokaryote, Taylor Woods and Hugh in their experiment wanted to show that it also works in eukaryotes, cells like plant cells or our cells. So they set up an experiment in this plant. I think it's called the broad pea, but they like to ask about it. Vivia faba is the plant that they use, and they showed in the chromosomes that it also has part of the old, and then if you label it the new, it's got the new in here. So you can see, even in real pictures, that part of it is labeled and part of it is not. And that's because part of it is the new stuff, and part of it's the old stuff. Semiconservative replication, and that is the Meselson Stahl experiment.